Hello everyone, welcome to my YouTube channel. In this tutorial, I am going to explain about two different concepts. The first one is ensemble, which is used in molecular dynamics simulation. So a molecular dynamics simulation can be carried out with different conditions, often referred to as ensemble. For example, in case of cell culture, you know, uh, we culture the cell outside of the body in a controlled environment. That means temperature, pressure or pH, everything is maintained. So different conditions are maintained. Similarly, for simulating biomolecules, we also maintain different conditions. Okay, so for simulation, we create a system with different ensemble with different degree of separation from the surrounding environment. Okay, so suppose we are simulating these atoms or molecules, and we create a simulation box to separate this molecule from the outer environment. Okay, and this separation ranges from completely isolated system, which is known as microcanonical ensemble, to completely open ones like a grand canonical ensemble. And the ensemble we choose depends on the specific problem we are trying to solve and the condition under which we want to simulate the system. Okay, so in this tutorial I am going to discuss about three mostly used ensemble in molecular dynamics. So let's start with the most basic one, that is NVE ensemble. So in NVE ensemble, the system has constant number of particles or moles, and the volume of the system is also constant or fixed, and total energy of the system is also constant. Okay, so it's a microcanonical ensemble, therefore correspond to an isolated system that cannot exchange heat or matter with the outer environment. But in this system, total energy is constant or fixed, but fluctuation in the potential energy and kinetic energies are still allowed, because the total energy within the system is actually sum of potential energy and kinetic energy. So this ensemble is known as microcanonical ensemble, and this ensemble is okay for simple system with a few number of molecules. Okay, but what if we want to simulate a complex biomolecules or more number of molecules like protein, DNA, these kind of molecules? So this system will be very complex. Okay, so suppose this is the system, and this system contain protein molecule, and here MD simulation starts from an unstable initial structure, and this structure and this atom within this molecule have high potential energy that need to be minimized. So when we minimize their potential energy of these atoms, what will happen? Because the total energy is fixed, so kinetic energy increases. Okay, when the potential energy is minimized, the kinetic energy increases because the total energy is fixed. As a result, finally the temperature of the system goes up, and when the temperature goes up, temperature increases. This may lead to several problems, like this thing may unfold, leading to unsuccessful experiments. So instead of NVE ensemble, we use NVT ensemble, where number of particle is fixed, volume of the system is also constant, but here the temperature is kept constant. In this case, system is allowed to exchange heat with the outer space, so that the temperature stays constant or fixed. Okay, so you can see heat can be exchanged in this case to keep the temperature constant. So it's known as canonical system. And to keep the temperature constant, here we use a thermostat. Okay, thermostat is one kind of algorithm which keep the temperature constant by allowing heat exchange with the surrounding environment. So how do the thermostat do it? The thermostat do it by simply scaling the velocities of the system so that we adjust the kinetic energy and consequently temperature. If the temperature is too low, the velocity of the atoms are increased and vice versa. So by increasing the velocities of atoms which are present within the system, the thermostat maintain the kinetic energy and consequently the temperature. Is known as canonical ensemble. The third ensemble is NPT ensemble. Okay. In case of NPT ensemble, the number of particle is constant. The, in this ensemble, pressure is also kept constant. Temperature is also kept constant. That means we have to use two algorithms, one for pressure and one for temperature. One algorithm maintain the pressure of the system and another algorithm maintain the temperature of the system. So in this ensemble, the system is allowed to exchange heat. At the same time, we can adjust the volume of the system so that pressure is kept constant. And this volume is adjusted by constantly rescaling the one or multiple dimension of the simulation box. This is the simulation box. So if we decrease the size of this simulation box, the atoms will come closer and the volume will decrease. Okay, that's how the volume can be adjusted and consequently the pressure of the system will be maintained. And you know, barostat algorithm is required to maintain the pressure of the system. So this ensemble is known as isothermal and isobaric ensemble because both temperature and pressures are maintained or kept constant. Okay. Now I am going to show you a few common thermostat algorithm. Okay. So first is Berenden thermostat is one algorithm which is used to maintain the temperature of the system. Next is nose hoover thermostat and the Bushy-Donadio Parinello thermostat, which is a modified Berenden thermostat, and in GROMACS software and in GROMACS molecular dynamic simulation package, it is referred as VD scale. So, so nose hoover and this VD scale, this thermostat are very commonly used in molecular dynamic simulation using GROMACS. But nose hoover thermostat is mostly used during the equilibration stage. Okay, and this VD scale thermostat can be used during both equilibration and also for production run. That means when we are actually simulate the biomolecules. And equilibration is a earlier step before the main molecular dynamics simulation stage. Okay. Similarly, barostat algorithms are also there, and Berenden barostat, which use similar kind of equation to maintain the pressure of the system, and then Parinello-Rohman barostat, which is mostly used in GROMACS software package and in other molecular dynamic simulation packages also. The second concept which I am going to explain is periodic boundary condition. Okay, so when we are simulating a system, so a simulation system contain about 100 to 1 million molecules. Okay, but a real system might contain many more than that. More molecules are present in real system, but in during molecular dynamics simulation, we simulate a very limited number of molecules. Suppose this is a simulation box. It's like square, but it actually in three-dimensional it will look like a cube. Oh, containing these molecules. Okay, and this single cube is therefore not appropriate to study the bulk property of the system. Okay, so periodic boundary condition is one kind of algorithm which is chosen to approximate the infinite system, that means a very large system. Okay, to approximate this infinite system, we use this small simulation box. That means a small part or a unit cell Here we consider this simulation box as a small unit, and using this unit we try to approximate the behavior of a very large system or infinite system. Okay, so in statistical mechanics there is a concept which is agrodicity. Okay, which tells us that infinite system at single time equals to single system for infinite time. That means if we are trying to assess the behavior of a system which is infinite, okay, then we can do that by simulating a finite system for infinite time. Okay, so if we use this small system for infinite time, if we create simulation box or same replica of this box in all direction of this simulation box. Okay, suppose this is the replica which are used in all direction of the simulation box. That means simulation box will be present in the center, and surrounding the simulation box the image or replica of the same simulation box will surround the central simulation box, and we do it to remove the boundary effect. That means when we do it, there is no boundary of this simulation system. Okay, why periodic boundary condition is used? So let's see. So what is the property of periodic boundary condition? In periodic boundary condition, small box are replicated in all direction. Okay, surrounding the central main box where the molecules are actually simulated. So there are no walls. So this simulation box becomes wallless, there is no wall, and actual simulation takes place only within this central box. Actually we are simulating, and in other box these are the image particles. And now if this particle suppose leave the simulation box, okay, then this particle from this simulation box will enter at the same time into the central simulation box. So when a particle leaves the box, on one side it is replaced by an image particle that enters from the other side. So total number of particles remain fixed within the central box. Now you may say, what will be the fate of this particle? It enter into the next simulation box, because there are infinite number of simulation box we are approximating, just to keep constant the particle of this central simulation box. Okay, wait it. So in this tutorial I have explained about two concepts, that means ensembles and periodic boundary condition. Okay, so in next tutorial I will be discussing a few other concepts. Okay, so that's all for this tutorial. Thank you for watching.